students. Today we are going to learn two topics, and that is a prokaryotic cells, in which we will learn a cell envelope and its modification, and then we will learn ribosomes and inclusion. In the prokaryotic cells, bacteria, blue-green algae, mycoplasma and PPLO is included. That means PPLO means pleuropneumonia-like organism. Okay, so these all group of organisms are included in prokaryotic cells. Okay, means bacteria, blue-green algae, mycoplasma and PPLO. They are generally smaller, multiply more rapidly than the eukaryotic cell because their structure is very simple structure, and that's why their multiplication method is very simple, and that's why speed or means they will divide rapidly. They may vary greatly in a shape and a size. The four basic shapes of a bacteria are a bacillus, that is a rod-shaped bacteria. Okay, suppose the organism is like a rod-like, then it is called bacillus. Then spherical, if the shape is a spherical, then it is considered as a coccus. Okay, then if coma shape organism is there, then it is called vibrio. Then if spiral shape is there, then it is called spirillum. So by this way, we can divide bacteria into four groups on the basis of their shape. If rod-like, that is a bacillus. Spherical, it is called coccus. Then coma shape, it is called vibrio. And if it is spiral shape, then it is considered as a spirillum. Prokaryotes have a cell wall surrounding the cell membrane. So suppose this is a typical prokaryotic cell. Surrounding this cell, of outermost layer will be of cell wall. Okay, and except mycoplasma, means in the case of a mycoplasma cells, it is not present. Means in the mycoplasma, cell wall is absent. Here, no well-defined nucleus. Okay, in this case, because this is a prokaryotic cell, we are talking about prokaryotic cell. So as we have discussed in last lecture, we have discussed that what is the difference between prokaryotic cell and eukaryotic cell. So there is a main difference in prokaryote and eukaryote is the nuclear membrane. Here in this case, nuclear membrane is totally absent, and that's why here no well-defined nucleus is there, means surrounding the nucleus there is a total absence of nuclear membrane. The genetic material is basically naked. Okay, and no envelope is found, or we can say not enveloped by a nuclear membrane. In addition to genomic DNA, here you can see this is the main DNA, that is called bacterial DNA. Along with this, means along with the main genetic composition, they will have some extra chromosomal DNA. Okay, many bacteria will have small circular DNA outside the genomic DNA. Here you can see in this diagram, this is the main bacterial DNA. Along with that, some extra DNA composition is also found, that is a circular in shape. Okay, so here you can see the circular DNA which is found outside the chromosome is called plasmid. Okay, so let me repeat. In addition to the genomic DNA, that is a single chromosome or a circular DNA, bacteria have small circular DNA outside the genomic DNA. Okay, and these smaller DNA are considered or called as a plasmid. Here you can see in the diagram, a blue color circular DNA is called plasmid. The plasmid DNA confers certain unique phenotypic characteristic to such a bacteria. So many characteristic will be due to plasmid, presence of a plasmid. In this slide, you can see the diagram, and these diagram indicate a typical prokaryotic bacterial cell. So let me go through this diagram in detail. So here you can see, first of all, cell is covered with the three layers. Okay, so innermost layer is a plasma membrane. Then out of these you will observe a cell wall. Then outermost layer is a glycocalyx, and that is also considered as a capsule. So total surrounding this prokaryotic bacterial cell, three layers are there. Innermost is the plasma membrane, middle one is a cell wall, and outermost is a glycocalyx. Okay, and out of this envelope you can observe 2 to 3 structures are there, and that is a pili. Okay, surrounding the cell there is a pili. Okay, then you can see this tail-like structure that is considered as a flagellum. Okay, so this is very important structure. And then if you observe very clearly or focus on this diagram, you can see this plasma membrane will see here. Okay, means this plasma membrane will show you one fold here. This fold-like structure is considered as a mesosome. Okay, this is very important structure we will learn in detail in this lecture. Okay, then here you can see some inclusion bodies are there. Then this is the plasmid, then ribosomes here you can see. Then all the structure which is found here is a cytoplasm. Okay, though all the structures are very important. In the center you can see a naked DNA or a naked genetic material is found, and that is called nucleoid. Okay, here it is not considered as a nucleus because it is not having a nuclear membrane, and that's why it is considered as a nucleoid, that is a main DNA component. So by this way we can say a cell envelope is consist of tightly bound three-layered structure. Okay, which we have already discussed in previous slide. The outermost is a glycocalyx, that is also called capsule, and it is followed by the cell wall, and then innermost is a plasma membrane. So these three layers will be found in the any prokaryotic cell, especially in case of bacteria. So bacteria can be classified into two groups. Okay, on the basis of differences in the cell envelope. Okay, in cell envelope you can see or observe some differences in between the bacteria. On basis of that you can divide the bacteria into two groups, and the manner in which they respond to the staining procedure which is developed by the Graham, that was the scientist, okay, Graham scientist, and developed by the Graham stain. Okay, so on basis of how it will be stained, it can be divided into two groups. Those that take up a Gram stain are considered as the Gram positive bacteria, and those which do not colored or do not are called Gram negative bacteria. Okay, means let me repeat, those that take up the Gram stain, okay, that are considered as a Gram positive, and then whatever the bacteria that do not take up any Gram stain, that is considered as a Gram negative bacteria. Now let me try to understand what is a Gram staining. So first of all you have to take some bacteria on the slide, which is a glass slide. Okay, then you have to apply a crystal violet, that is one type of a stain, and that is a purple in color. It is a purple color dye, and on application of a crystal violet you will see a bacteria will be observed a purple in color. Then in that slide you have to apply iodine, that will be act as a mordant, that is a helper molecule. Okay, and it will be applied on this slide. Then in next step you have to just wash out the slide with the help of alcohol, and that is called decolorization. As a result of that, here you can see a slide will be decolorized. Okay, and then in the next step, in the last step, you can see you have to apply safranin, that is a counter stain or second stain. Okay, so after these all staining, you can see here what happened inside under the microscope, what you can observe. So during application of crystal violet, you will see all the bacteria will be stained violet or purple in color. And in the next application, you will see same result. Then on the third step, if you observe, all the bacteria which are not gram staining, means the gram negative bacteria, the gram negative bacteria will be decolorized over here, but a gram positive bacteria remain as it is. So gram positive bacteria will be colored as a purple, and it will be observed as a purple in color, whereas the gram negative bacteria you cannot observe because they are now it is colorless due to alcohol wash. And finally you have to apply safranin. As a result of that, what happened, whatever the bacteria is gram positive will be look after under the microscope, you will observe that bacteria will look purplish color, and that is considered as a gram positive bacteria because it is stained with the help of crystal violet. Okay, and the bacteria which is colored with the help of safranin and look like a pinkish color, and it is considered as a gram negative bacteria because they cannot be stained with the help of a crystal violet, and they will be decolorized in presence of alcohol. Okay, so by this way we can divide organisms or bacteria, this prokaryotic organism or a bacteria, into two groups. Mainly bacteria, gram negative and gram positive bacteria. Now let me try to understand more. Here you can see a cell wall of a gram positive versus gram negative bacteria. So both the cell wall is you can see here. Okay, gram positive bacteria cell wall will have a lipotecoic acid. Okay, here you can see some molecule, it is a lipotecoic acid on the surface protein and teichoic acid. Okay, this is the peptidoglycan layer. Okay, the cell wall which is made up of one specific component, and that is a peptidoglycan layer. And then very common cell membrane is there, which is made up of phospholipids. This is the cell wall of a gram-positive bacteria. But here in this case, in gram-negative bacteria, why gram-negative bacteria and gram-positive bacteria stain differently? Okay, that all bases on the cell wall, a type of a cell wall. So here peptidoglycan layer is there. Okay, and here you can see gram-negative bacteria where lipopolysaccharide layer is there, that is extra layer, and surface protein. Here you can see lipopolysaccharide layer is not there. Okay, then lipoprotein is there in between, and then peptidoglycan layer is very thin layer, whereas here thick layer is there. Okay, then cell membrane. So this is the basic differences between gram-negative and gram-positive bacteria, and that's why their staining mechanism is different. So this is the basic difference between gram-negative and gram-positive bacteria, and that's why they will stain differently. So now let me summarize. Glycocalyx or a capsule differs in composition and thickness among different bacteria. Okay, it could be loose sheet called the slime layer. If it is in loose form, then it is considered as a slime layer, while in other it may be thick and tough, and if it is tough like structure, then it is considered as a capsule. Okay, so glycocalyx may be of two kinds. Loosely formed, it is a slime layer, and if it is tightly formed, then it is called capsule. The cell wall determines the shape of the cell. Okay, and it provides strong structural support and to prevent the bacterium from the bursting or collapsing. This is very important structure. Cell wall is okay. The plasma membrane is a semi-permeable. Means what is the meaning of a semi-permeable? They only allow several molecules to be come in or out. Okay, that's why it is called semi-permeable in nature and interact with the outside world. The membrane is similar structurally to that of eukaryotic. Almost it is similar to the eukaryotic After cells. Learning about envelopes, that is a glycocalyx, then cell wall and the cell membrane. From inner side, if you look inner side, there are several structures found in that. And first important structure is a mesosome. Okay, a special membranous structure is called a mesosome, which is formed by the extension of a plasma membrane into the cell. Here you can see it is actually a cell membrane extension. Okay, inside the cell. Then these extensions are in the form of a vesicle, tubule or lamella. It is different in different cases. Okay, and what is the main functions of a mesosome? It is very important for the cell wall formation, then DNA replication and distribution to the daughter cell. Okay, respiration, secretion process is also that is responsible by, we can say mesosome is responsible for that, to increase the surface area of the plasma membrane and enzymatic content. Means mesosome play a very important role in case of a prokaryotic cell. If you observe any prokaryotic cell, we can divide them into two groups on the basis of their motility. Okay, so bacterial cell may be motile or may be non-motile. If they are motile, they have some thin filamental extension from their cell wall, and that is called flagella. Okay, if motile bacteria only possess the flagella, then bacterial flagellum is composed of three parts. Here you can see, and first part is a filament, then a hook and basal body. So here you can see in the diagram, okay, this greenish color tail like structure is a filament. Okay, then his bluish color is a hook region. Okay, and then all the parts, that is a L ring, P ring, and that all is called basal body. So by this flagellum is made up of three parts: filament, hook and basal body. Okay, the filament is the longest portion. Here you can see filament is the longest portion and extends from the cell surface to the outside. Along with the flagella, some other structures are also there which are responsible for the motility of a prokaryotic cell, and that include pili and the fimbriae. Okay, let me understand more. The pili are elongated tubular structures and it is made up of special kind of a protein. Okay, and the fimbriae are a small bristle-like fiber sprouting out of the cell. Here in this diagram you can see surrounding the cells some microstructures are there. Okay, brush like structures are there and it is called fimbriae and the pili. Okay, the fimbriae are small blister-like fibers sprouting out of the cell. In some bacteria, they are known to help to attach the bacteria to the rocks also, and in the streams, and also on the host tissue. So this is very important to create any disease in the organism. Okay, in the host organism, they are responsible for causing the disease in the organism or host organism. Now let me start a second topic, and that is ribosomes. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. As we have learned that in previous lecture, we have already learned ribosomes are found in both the cells, whether it is eukaryotic cell or a prokaryotic cell. But when you are studying about the prokaryotic ribosome, it is attached with the plasma membrane. Okay, they are about 15 nanometer by 20 nanometer, okay, in size, and are made up of two subunits. Okay, it is made up of two subunits. Here you can see one larger and one smaller. Okay, and it is combinedly, it is composed of 50 As. S means Svedberg unit. We will learn in detail in next slide what is the Svedberg unit. 50 As and 30 As. The larger unit is a 50 As and the smaller unit is a 30 As. That S indicate the Svedberg unit, which when present together form a 70s. When they both the larger and smaller fragment when bind together, it will form 70s, means 70 Svedberg unit of a prokaryotic ribosome. So here you can see the comparison in between prokaryotic ribosome and eukaryotic ribosome. So here you can see a larger fragment of the prokaryotic ribosome is of 50 As, 50 Svedberg unit, whereas the smaller one is a 30 Svedberg unit, and if it combine together, it will be form 70 Svedberg unit of a ribosome. Then in case of a eukaryote, you can see a larger one is having a 68 subunit. Okay, whereas a smaller one will have 40 S subunit, and if it combine together, it will form 80 S subunit. Now here in this slide, let me try to understand what is a Svedberg unit. Okay, so Svedberg unit is the measure of how quickly particles sediment in the ultracentrifugation. So this particle size is defined by using ultracentrifugation. That is one method by using which you can define the centrifugation charges. Okay, and it will indicate it as a Svedberg unit. Okay, larger the particle, the greater the S value. Here you can see in this diagram, this red color is a larger molecule, right? So it will sediment quickly as compared to this one or this one. This is the largest one, red is the largest one, this is the middle one, and this is the smallest one. You can see which one is get sedimented first. A red one is sedimented first, means larger the molecule, it will be sedimented first. Okay, so if larger the particle, the S value will be also large, means larger the particle, Svedberg unit will be larger. Okay, and smaller subunit of the ribosome sinks slower. Here you can see yellowish color unit is a small, and that's why it will sink slowly. Okay, as compared to these two. Okay, and that's why their subunits will be, means Svedberg unit will be very low. So smaller subunit of a ribosome sinks slower than the larger subunit, and by this way, on the basis of how fast it will sink, okay, on basis of that only, that Svedberg unit will be decided. Protein synthesis is very important process in any organism, and that process of a protein synthesis occur with the help of ribosomes, or ribosomes are the site of a protein synthesis, we can say that. Okay, and sometime what happen with the mRNA? mRNA is a messenger RNA which will be passed from the DNA which is in the nucleus. Okay, DNA will form mRNA, and on the basis of mRNA codes, that protein synthesis will occur on the ribosomes. Sometimes what happen? Several ribosomes, means more than one ribosome will attach on the single mRNA. Here you can see a bluish color strand is a mRNA, on which you can see smaller subunit and larger subunit. That ribosome will attach on the, so many ribosomes will be attached on the strand together. Okay, and if so many ribosomes attach on the single mRNA, that is considered as a polyribosome or a polysome. Okay, so what is the definition of a polysome? Several ribosomes, means so many ribosomes is bind on the single mRNA, then it is considered as a polyribosome or a polysome. The ribosome of a polysome translate the mRNA into protein. So this is a central dogma. The DNA in the center will pass message in the cytoplasm in the form of a mRNA. That process is called transcription. After the transcription, this mRNA will be coded for some message, okay, for the protein synthesis, and on the basis of the strand, okay, on the codes which are present on the mRNA, according to that protein synthesis will occur, okay, and that process by using mRNA, whatever the protein synthesis occur, that process is called translation, okay. That's why the ribosome of a polysome translate the mRNA into protein. Another topic is the inclusion bodies. So let me try to understand what is the inclusion bodies. Reserve material in a prokaryotic cell, okay, which is unused material, okay, will be stored in the cytoplasm in the form of inclusion body. So what is the inclusion body? Let me repeat. Reserve material, okay, in the prokaryotic cells are stored in the cytoplasm in the form of a inclusion body. These are not bound by any membrane system and lie free in the cytoplasm. Here you can see purplish color structures. Okay, dot-like structures are an inclusion body, and that will be lie freely in the cytoplasm. And such inclusion bodies may of so many kinds. Okay, maybe your phosphate granules, maybe cyanophysin granules, then sulfur granules, then glycogen granules, polyphosphate granules. It is depend on which type of reserve material is found in the cytoplasm. Okay, then in some organism gas vacuole is also found. Okay, here you can see this is the gas vacuole, and this gas vacuole is very important, okay, in the aquatic type of bacteria, because it will help to float on the water. Gas vacuoles are found in the blue green and purple and green photosynthetic bacteria, because these photosynthetic bacteria are found in aquatic environment, okay, and these bacteria have to swim around the water, and for that purpose that gas vacuole is very important. This is your homework for today's lecture. Just you have to write down in your notebook. Next lecture, we will learn next topic, and that is cell membrane. So till then, goodbye.